Hi, my name is Ashley,
Hi, my name is Destiny.
and we were given figure 6, A through I, for the journal article Senescence Reprogramming by TIMP1 Deficiency that Promotes Prostate Cancer Metastasis.
So, our big question for the article was, what is the molecular function of metastasis prostate cancer? Some of the relevance and significance we got was that it's a silent killer, a silent killer is basically prostate cancer that 1 out of 8 men experience, and not a whole lot of symptoms are shown until the cancer spreads and grows silently. It grows in the urinary glands from months to years. When the TIMP1 gene is removed from the mice model, the pro-metastatic process proceeds to spread. Our goal, or the goal for the journal article, was to determine roles of tumor senescence.
All right, and for our figure, we have figure 5, which was shown from previous figures. And to take away from this figure, it is to determine the mechanism by which the senescence contributes to this aggressive tumor. This experience is usually used in mouse embryonic fibroblasts and performed in proliferation. Senescence is also involved, cell invasion, and migration assays. So this is our research question. With the research question, we were given figure 6, A through I, and it is titled TIMP1 is lost in advanced and metastatic prostate tumors due to promoter methylation. And with these experiments that we were researching, we came up with a question where our question is, how does methylation affect the TIMP loss and promote metastatic prostate cancer?
Okay, so these are both figures A and B, as you can see right here. So for the research of figure A, it is experimenting the TIMP1 staining using prostate cancer tissue microarray, whereas figure B is calculating the highs and the lows of the TIMP1 levels. The results of figure A and B is that we see benign hyperplasia has more staining on the far left of figure A, which indicates that we have higher levels of TIMP1, and which is shown in figure B. This shows that without the alteration of any gene or protein, the TIMP1 gene is at work. In primary tumor, there are primary cancer cells that already exist in the body. With the decrease of TIMP1 levels, there is less staining demonstrated, which is demonstrated on the middle left of figure A. With metastatic tumor, the low levels of TIMP1 are at 96%. With the loss of the TIMP1 gene, we don't have any stains on the metastatic tumor. In the diagram for figure A, it shows that the metastatic tumor has surfaced and spread with no TIMP1 gene.
All right, for figure C and D, they were both quite similar. Figure C and D is an RNA sequencing analysis of the prostate tumor areas of TA1 and TA2, which is also described in red and black. And normal prostate tissues showed decreased levels of TIMP1 in the tumor areas as presented in figure D. The results of figure C was on the bar graph, there are TIMP1 log2 ratios of the RNA sequencing that counts between tumor areas, which is TA1 and TA2, that are compared with those in normal adjacent tissues within the same tumor samples. And on the right is figure D, which is the pie chart that represents the quantifications of TIMP1 levels as shown in also figure C. The fraction in the black represents the normal levels of TIMP1, that in red represents the negative tissues of TIMP1, while the gray, which is 5%, is positive tissues of TIMP1. And as for figure E, Figure E is basically just describing the promoters with the tumor gene and how it shows high levels of methylation, whereas normal like promoters show low levels of methylation. And just wanted to be clear, methylation is promoter regions of the body that can silence the tumor suppressor gene, which can lead to other mutational events and develop numbers of cancers. Based on this graph, the promoter is on the x-axis while the methylation is on the y-axis. Promoters with the tumor gene show high levels of methylation, whereas normal like promoters show low levels of methylation. This determines that methylation is involved in tumor genesis.
Okay, so now moving on to figures F, G, and H. So for the research portion, figure F is determining the significance of the positive correlation of P10 and TIMP1. Figure G is using tissue microarray to determine the protein expression in cancer, normal and tissues. Figure H is the percentage of cases in P10 and TIMP1 protein in 2 tumor samples. The results of figure F is that this bar plot enlists prostate cancer datasets demonstrating a positive correlation between the expressions of TIMP1 and P10. Figure G is representing images of human TMA, which is tissue microarray, and refers to the practice of relocating multiple tissue sections, samples stained for P10 and TIMP1 in primary and metastatic tumor cases. There is more staining in primary tumors than there is in metastatic tumors for P10 and TIMP1. In Figure H, this bar plot represents the percentage of cases with P10 and TIMP1 protein expression in primary and metastatic tumor samples. In primary tumors, there is a big result in having high levels of P10 and low TIMP1 levels. In metastatic tumors, there is low P10 and TIMP levels and barely low P10 and high levels of TIMP1.
All right, for figure I is TIMP1 and P10 determine how they react in low and high levels of methylation. Methylation can change the activity of a DNA segment without changing the sequence. TIMP1 shows high and low levels of expression on the heat map with high methylation, and it is reported that low levels express more than high levels of TIMP1. As you can see, the red high expression levels of TIMP1 work very well with low methylation TIMP1. As you can see that the red is more prevalent and occurs the most in TIMP1 low methylation than it does in TIMP1 high methylation. Vice versa also applies to low expression levels, which is colored in green. For the next one, which is P10 expression, P10 shows high expression levels of TIMP1 low methylation and shows low expression levels of P10 on TIMP high methylation. As you can see, P10 high expression levels is in pink and P10 low expression levels is also in purple. The purple low expression levels is more prevalent and occurs the most in high methylation, whereas the pink also shows more and occurs the most in low methylation. And last but not least, P10 deletion and mutation. With the definition of methylation that we all know, high methylation can alter DNA activity, so there is various reportings of deletion and mutation in TIMP methylation, high methylation. No alteration occurs in low TIMP1 methylation because there's no DNA activity for methylation to disrupt. So no alteration is colored in gray, and deletion and mutation is also colored in black. And it is more prevalent and more shown in TIMP1 high methylation, as you can see on the blue side of the heat map bar. And you can see it also on the low methylation on the heat pad.
Okay, so after reading the article, we came up with two questions. First one being, if performed on humans, how would TIMP1 gene deficiency affect humans compared to mice? And the second question we came up with was, what would happen if human genes if they played a role in preventing metastasis without TIMP1 gene?
Right. And to better have a better understanding of that, how would TIMP1 gene deficiency affect the body of humans compared to mice? We asked that question just because mice and humans also have basically the same genetic like makeup or have like the same genetic code, I believe. And so we are able to use mice models to emphasize like drug treatments or medication to be either performed on us, but we obviously use mice models to help benefit us. And so with that question, I am wondering how would the TIMP1 gene deficiency would affect on us if that gene was removed, which also follows up with the second question is what would happen in human genes if they played a role in preventing metastasis without the TIMP1 gene? Like what other genes would like come and prevent those um, metastatic cells or the metastatic prostate cancer cells from spreading so quickly? That's like the one question that we had both came up as we started researching and figuring out all the figures and experiments of this certain figure of figure 6.
Information that wasn't told in the article as well.
Yes.
So here are references that we used for our research, and this brings us to the end of our presentation. So we just want to say thank you so much for listening, and we hope you have a great day.
Yes. Thank you. Have a great day.